Hello, everyone. My name is Evan Pogue, and I'm a sophomore undergraduate researcher in Dr. Uze Amir's lab. Today, I'll be presenting about fast in vivo sodium imaging and T2 star mapping using non-localized 2D FID MRSI at 3 Tesla. Today, I'll be presenting about the importance of sodium in skeletal muscles, give an introduction into the basics of MRI and sodium MRI, talk about the purpose of Amir research, provide my proposed methods and materials, and give my results and conclusions, as well as talk about my ongoing experiment using physiological interventions for our new proposed MRSI method, as well as provide my future directions. To begin, sodium is one of the most important elements in our body, and it plays a major role in our muscles. It assists with voluntary and involuntary muscle contraction and improves and maintains nerve impulse transmission. A lack of sodium in the body leads to muscle fatigue and cramps. After placed inside a powerful magnet, such as an MRI machine, the protons in our body align with the magnet and they begin to precess. A radiofrequent pulse, or RF pulse, is turned on that allows the protons to spin. Once the radiofrequent wave is turned off, the protons that were spinning in phase will no longer spin synchronously. This is known as dephasing. Dephasing in T2 relaxation is extremely fast, and when the RF pulse is turned off, transversal magnetization will be lost. So as you can see on the image on the bottom right, the protons are aligned with the magnet, and then once the RF pulse is turned off, they begin to dephase. And once they completely dephase, the transversal magnetization will be lost. T2 is the time needed for the protons to dephase up to 37% of its original value. Each tissue has its own T2 relaxation time and curve, which allows for contrast in the MRI image. As you can see on the graph, fat has a shorter T2 and will dephase quicker than water, and thus the MRI can show us contrast between fat and water. In addition, the protons must be in phase for the coils to receive the signal. The purpose of the Amir lab is to implement an accelerated MR acquisition method, allowing to map sodium T2 star relaxation in absolute concentration within skeletal muscles at 3 Tesla. Now, some of you may be asking what T2 star is. Well, T2 star is an observed or effective T2 when the transverse magnetization decays much faster than predicted. For this reason, T2 star is always less than or equal to T2. My contribution to this project was to first learn the basics of MRI, analyze the results, and use this MRSI method for future projects, which I'll explain about in later slides. Compared to signals for hydrogen protons, sodium has a very low signal, about 9% of the NMR sensitivity of hydrogen. In addition, sodium decays bi-exponentially and extremely fast, so therefore it is very hard to pick up its signal. To overcome these issues, one needs to use the undecayed signal, which can be estimated by measuring it as T2 star values. We need to start measuring as early as possible and measure enough data time points of the decaying curve for better fitting quality. The density-weighted concentric rings K-space trajectory is used to boast the signal trend ratio. Since conventional slice selection techniques result in a long acquisition delay, we are proposing a pre-excitation technique called SIRENE, which stands for single-shot inversion recovery-based non-echo. Instead of targeting the slides of interest, the gradient will be applied to target the area outside our slides of interest and suppress it by applying inversion recovery pulse and OVS, or outer volume suppression band, which allows for a shorter acquisition delay. Alternatively, to achieve a shorter echo time, 3D MRI can be used. It has an advantage of providing 3D spatial information. However, it detects fewer time points within a longer scan time compared to our proposed MRSI method, which requires 64 time points within only 15 minutes, which is a really short scan time. In this study, we used a dedicated sodium transmit knee coil. For quantification, we used 4 calibration bottles filled with known sodium concentrations. As you can see on the image on the top right, this is our sodium-only transmit/receive birdcage knee coil, and on the bottom photo, we have our 4 calibration phantoms. For quantification, we used 4 calibration bottles, and copper sulfate was added to each bottle to mimic the in vivo T1 relaxation time. The MRSI was prescribed with 2.5 by 2.5 spatial resolution and 64 time point measurements. To evaluate the spatial mapping of the MRSI, we added a 3D MRI as imaging reference. After making sure the siren works properly, an in vivo study on the calf region was performed. The MRSI data was reconstructed as follows. First, the data underwent regrading and reconstruction. Since we acquired multi-data points over time, a beta-knot homogeneity mapping is possible and was used to correct the data. Following, the data was denoised using low-rank approximation before performing the T2 star mapping and correction. Finally, the absolute sodium mapping was performed by calculating the signal to the known reference concentrations. As you can see, a considerable enhancement in sodium concentration is gained after T2 star correction that can be seen from the difference map. In conclusion, the proposed MRSI method is a reliable method to estimate absolute sodium concentration and relaxation time constant, or T2 star information. For more information on this experiment, see the attached link. Next, I wanted to challenge the sensitivity of the MRSI method from this research project to evaluate sodium concentration in the gastrocnemius muscle through physiological manipulation. We expect to see an increase in sodium concentration in the gastrocnemius muscle after physiological manipulation, which is our hypothesis. The materials and methods for this project include our newly acquired MRSI method. This is the same MRI used at Purdue's MRI facility, as you can see on the image on the top right, where we actually performed our experiment. We scanned the right gastrocnemius muscle once before and once after physiological manipulation. Afterwards, we used MATLAB to reconstruct the MRI data. To reconstruct the data, we selected 10 voxels from the brightest phantom and 20 voxels from the skeletal muscle tissue for both scans. We inputted these voxels into MATLAB to find their complex numbers. Once we found the complex numbers, we computed them into Excel and found the mean values. We then took the ratio of the mean values from the first scan to the second scan. The first scan meaning before physiological manipulation, and the second scan meaning after physiological manipulation. We expect the ratio to be less than 1, since we expect to see an increase in sodium concentration for the first scan. Highlighted in red is where we took the voxel data from. The ratio of the mean from the first scan to the second scan was greater than 1 for the skeletal muscle tissue, while for the phantom bottle, the ratio of the mean was less than 1. This indicates that there was a gain in sodium concentration for the phantom bottle and a loss in sodium concentration for the skeletal muscle tissue. Although the new method successfully measures sodium content, results are not in line with our expectations. The image on the left shows the first scan, and the image on the right shows the second scan, before and after physiological manipulation. Our preliminary data shows that our new method is working, as we are able to produce 3D sodium muscle content. In the future, we would like to rerun this experiment to figure out why these ratios do not have the same trend. In addition, we would like to explore whether this method is sensitive enough to detect physiological interventions such as dietary intake or physical exercise. Here is a list of my citations. Lastly, I would like to acknowledge Dr. Uze Amir for being a great mentor throughout my research, as well as his 2 PhD students, Xin Shen and Nicholas Farley, who assisted in teaching me how to use MATLAB and reconstruct MRI data. I would also like to thank Andres Morales for helping me learn MATLAB as well. And lastly, I would like to acknowledge Dr. Freeman for allowing me to be a part of Purdue's undergraduate honors research program. Thank you.